Hi, my name is Sam Lohse. I'm an environmental and analytical chemist here at Central Washington University, and I'd like to tell you a little bit about the research going on in our research group. In the Lohse research group, we look at the chemistry of emerging materials contaminants. So materials contaminants are microplastics and nanoparticles. These are different from traditional molecular chemicals that become environmental contaminants because they have specific properties that are the result of their size and shape. They exist on an odd length scale, most of them being somewhere between the size of a very large protein and kind of an average size virus, which means they can get into compartments, they can get into organs, tissues, even organelles inside of cells in ways that traditional chemicals can't. They can bypass active and passive transport systems to permeate cell membranes. And as a result, we have now an emerging technology where we want to take advantage of the surface area, the size-dependent properties of nanoparticles and microplastics to facilitate applications that we couldn't realize using traditional chemicals. These are exciting, game-changing applications potentially. Everything from what we would call theranostic agents in biomedicine, materials that can both assist with diagnosis and deliver site-specific medicine payloads to tumors or open up new avenues for cancer therapy beyond traditional surgery and chemotherapy. But we are now opening the door on the use of chemicals in these applications that don't play by the same rules that molecular chemicals and our traditional environmental contaminants do. So while nanoparticles and microplastics have exciting next generation applications associated with them by virtue of their size-dependent properties, the fact that they have unusual size-dependent properties also opens up the possibility that they pose unique hazards to human and environmental health. So in our research group, we go down two main avenues where we try to understand the connection between what we call the physiochemical properties of nanomaterials, the size, shape, and chemical composition of the material, and the biological or ecological effects of the nanoparticle. And we also investigate the development of low-cost portable sensors to detect emerging materials contaminants in the environment. Specifically, in terms of the path of the nanoparticle through an animal's body or through an ecosystem, we are interested in characterizing how the nanoparticles or the microplastics are transformed by their interaction with other biopolymers and macromolecules that are in that environmental or that biological medium. So, for instance, nanoparticles, when they enter an animal bloodstream, often develop a shell of adsorbed proteins, what we call a protein corona. And so rather than a nanoparticle moving through your bloodstream, what your body sees is a kind of virus-sized particle that's covered in an overlayer of serum proteins. And that has consequences for how the nanoparticle is recognized and interacts with your immune system, and whether you can actually design a nanotherapeutic that will deliver a chemotherapeutic agent directly to a cancer site or bind specifically to a tumor. These type of interactions also happen in the environment, where you have the formation of ecocoronas, where nanoparticles interact with natural organic matter and other carbohydrate polymers and components of bacterial biofilms that overcoat the nanoparticle, sometimes gluing multiple particles together or trapping other environmental contaminants inside the outer boundary of the nanoparticle itself. This means that potentially as nanoparticles move through the environment, they could act as a kind of transport vector, delivering high concentrations of more toxic materials contaminants into an organism's body. And so we try to make a connection between the original chemical composition and physiochemical properties of the nanoparticle and the biological or ecological identity of the nanoparticle after it's undergone some of these transformations to better predict the fate and transport of the nanoparticle through either an ecosystem or an organism's body. And we also work with collaborators at different institutions to understand how the transformations impact the toxicity and the genomic response of organisms that ingest nanoparticles. Students working in our research group gain expertise in nanoparticle synthesis, nanoparticle characterization, biochemical assays, basic studies of colloidal aggregation and transformation in the environment. We work with a variety of instruments on campus, everything from absorbance or fluorescence spectrometers to the scanning electron microscope in the Murdoch lab, various light scattering instrumentation, including the master sizer on campus. We do interdisciplinary research, and we look at the fate, transport, and characterization of nanoparticles in many different areas of the environment from a lot of different perspectives. Our work is inherently interdisciplinary, and we like to have students who are chemistry majors and students who are majors adjacent to chemistry participate in our research. We also have a new area that's opening up where we're looking at developing portable colorimetric sensors to detect, identify, and quantify nanoparticles and microplastics that enter freshwater systems. If you're ever interested in talking about research, I encourage you to drop by anytime. You can reach me at my CWU email address, samuel.lohse@cwu.edu. Also give me a call or just stop by the office. I'd love to talk to you about research.